I'm just gonna apologize ahead of time since this one's a little long, all right? A little longer than my normal presentation. So we're gonna talk about the function of the cell membrane. There are four main types of passive. We're gonna talk about passive transport first. Passive transport means that it doesn't need energy to occur. It's actually going to move according to what's called a concentration gradient. It's going to go from an area of high concentration to low concentration. I always think of it as like you're in a hill and you're sitting on top of a hill on your bike. I'm a terrible bike, but you got my point. And you coast down. So you can think of it as high elevation to low elevation. Okay. And once you start going over, you know, the hump of the hill, you go down due to, gravity. So that's kind of what's happening here. You don't need energy for these types of movement of materials in and out of the cell. So like I said, diffusion, area of high concentration to low. It does not require any energy. So it's going to start off, say, perfume's going to start off, if you release it here, it's going to spread out throughout the room. We can also look at it in terms of the cell membrane, right? If the cell membrane is what we call selectively or semi-permeable, means some things can go through. They don't need help. They can zip right in and out. So right here we have a basic cell membrane. And when I talk about a concentration gradient, you can see we are highly concentrated up here. So we have a high concentration on that side and a low on this side. And this bar is showing like over time. So as we progress from left to right, time has advanced. And so we're going to go downhill and more is going to go this way until eventually we have equal amounts on both sides. That's referred to as dynamic equilibrium or just equilibrium. Now, this does not mean these solutes are not moving anymore. It just means that we have approximately the same moving this way as we have this way. So here's our dynamic equilibrium. You can see the molecules moving back and forth. We're trying to get equal number on the same side, but even when it has equal numbers, we're still having movement from right to left or left to right. Okay, so diffusion is when we talk about any other substance that is moving from high to low except for water. Water is so special, it gets a special name. The diffusion of water is called osmosis. And the movement of water will also move due to these differences in concentration gradient. There are three types of solution we're going to talk about: hyper, hypo, isotonic. I did talk a little bit about this in class. A little solution review. So remember, a solution is a mixture of two substances. It could be the solute is what you're trying to dissolve. So if you're trying to dissolve sugar into your coffee, the solvent is what you're dissolving in. And even though you call it coffee, it is basically flavored water. Okay. Like I said, there's three types of solutions. We have hyper, hypo, iso. I always think about hyper as more solute. A solute to think about is sugar. If you give a kid too much sugar, they become hyper. And then I like to jump down to this one, isotonic, equal. Like an isosceles triangle, equal on all three sides. Equal means that you have reached dynamic equilibrium. You have the same amount of solute on both sides of your cell. Hypo means you have more solvent. Okay, so you can think of this as higher solute. This has less solute, okay, in the solution. In a hypertonic solution, because there is more of, let me change color. Ooh, let me do this. Just start over here. Because there's more solute, we'll say salt is on the outside, because we're talking about the solution that the cell is sitting in. That means the water is going to exit the cell and try to dilute. I always use that term. You want to dilute the solute. Dilute the solute. We can't move the solute in and out of the membrane, but we can move water. And so we can see what happens with these blood cells. They're becoming dehydrated and shriveling up. In plant cells, we call it plasmolysis. So again, if we have all of this solute on the outside, we have our nice and turgid cell here. And over time, because we're losing water to dilute the solute, you can see that the membrane here, the cell membrane is pulled away from the cell wall, and that is referred to as plasmolysis. So you can see in this picture here, all this empty space, okay, which was occupied by like the cell membrane would have been. This one's not as bad, but the cell membrane would have been fully around, pushing against the cell wall and occupied by chloroplasts. These are all chloroplasts. They've all been gathered up and it's kind of sucked into the center. Notice the central vacuole gets very, very reduced inside. So everything kind of gets pulled into the middle of the cell. Hypotonic environments, that's where you have more solute. So let me draw. We have a lot more. Oh, hold on, let me draw something better. We have a fishy. Draw my little fish. Okay. Now, most freshwater organisms have a problem with this. Okay, so we have freshwater bodies around here. So when we say that, what we're saying is that there is less solute outside of my little fishy and a lot more inside. Okay, so the water is going to want to go in. What didn't change color? The water is going to want to go into the fish, and eventually the fish would explode, and that would be a problem. Okay, so this is a problem with organisms that don't have cell walls. So we have to figure out a way of getting pumping the water out or pumping the solutes out. We've got to equalize this difference in concentrations of water and solute. Plant cells not only deal with this well, but they like it because it makes them nice and really, really, really what we call turgid or full, and that helps keep them nice and upright. All right. So that cell wall will literally, the cell membrane will push against the cell wall and kind of say, okay, we've got enough, we're full or full. And then as soon as we start using that water and photosynthesis and stuff, then the cell membrane is not as tight and we'll get more water in there. Now, like I said, animal cells, they're going to have a problem. What's going to happen is you're going to have that water going into the cell until it bursts. You do not want your red blood cells to burst. If they do, you cannot carry oxygen. Game over, you're dead. Right? Which is why in the nursing health profession, they have to be very careful with what IVs they're sticking into your blood. It needs to be in what we call a ringer solution. And what that ringer solution means is it's isotonic. All right. So we're not going to mess with your solute solvent concentrations, which is good. If a cell cannot, like we said, it's going to burst. What prevents a plant cell from bursting? I kind of talked about that. And that would be, again, the cell wall. Now, I've used that word turgor pressure or turgid. Okay, or turgid, which means it's nice and full. And you can see as we've got this central water vacuole pushing out as it's full with water on to the sides of this plant cell, which should be a rectangular box. I understand it, but I did like what it was showing in this picture. Okay, you can see the organelles are kind of all in the space between the central vacuole and the cell membrane. So we have a high amount of, basically it's like water pressure. And that water pressure keeps your plants nice and upright. Because remember, plants don't have bones. They basically have the cell walls and they have that turgidity, the turgor pressure, right? So here's a side-by-side comparison. Let's just start with our isotonic. Notice we have water going in and out in equal amounts. We have our hyper, I'm going to use red, hyper, which means we have more solute on the outside, and so water's going out to equalize. It's going out to dilute the solute. And in this case, we have more, that didn't show up very well. We have more solute inside less on the outs. That's the hypotonic solution. Hypo means less solute. Hyper means more solute in your solution. And this means the same. So again, in this case, we're going to try to dilute the solute. So that means water is going to be taken into the cells. So how cells deal with osmosis? Well, freshwater organisms, they could dissolve more solutes than the water. So we're going to have a lot more solutes in that cytoplasm. So the water would be hypotonic. So which way would the water move? The water would move into the cell. So paramecium have developed a pump, okay, called a water contractile vacuole. So they have a special organelle, and it is constantly pumping water out of the cell. The thing with this is it does require ATP. Think about it as your sump pumps and stuff in your basement, right? If you get heavy amounts of rain, water comes in, you have to use electricity to pump it out. The power goes out, you got a problem, right? You got water that's going to be coming into your basement and flooding your house. That's not good. So here is our paramecium. It has right here this water contractile pump, and you can see here it's full, and then it releases the water outside the cell, and here it's empty. Now, multicellular organisms have organs, right? Kidney systems that will help to get rid of all that extra water that it's taking in. So, like if you've had a freshwater tank, aquarium, you know you have to be careful with the ammonia levels because those fish are constantly basically peeing that water out. And then when they break down proteins that has ammonia in it, or amino, is we're going to take those aminos out of our amino acids, out of our proteins when we break them down in a process called deamination. Don't need to know that unless you're taking biochem. But and so that basically, as they excrete that ammonia into the water as they're constantly peeing, which is why if you get a freaking goldfish at a fair, or your kid does, and you're like, oh dear Lord, and you kill it within days, that's why goldfish are especially dirty. So you almost have to change the water like every day, unless you put them in an actual tank. And even then, you have to be careful because they just secrete so much of their waste products that they basically start to burn up. The pH changes on it, it becomes like base, and they start to burn. They basically will burn themselves up in your tank. Okay. And then you go and find the fish floating, and your kids are all upset. Not good. Not good. I digress. All right, facilitated channels now, they are there to help these larger molecules cross over the membrane. All right, so there's two different ones we're going to talk about. We're going to talk about a channel protein and a carrier protein. They are going to work from high to low concentration, right? So they do not require energy. And so notice we have a high concentration outside, and it's going to let it flow inside until we have the same concentration, or ISO, or you can say it's an isotonic solution. And we could do the same thing with a carrier protein. So a carrier protein is going to give it a little bit more love, right? It's going to hold on to it, change shape, and then bring it into the cell. All right. Now we've been talking about transmembrane. So channel proteins are a transmembrane, right? They go all the way through. They allow the passage of molecules that are too large to go, right? These cannot go through. They have to have a passageway in. And again, passive because they rely on concentration gradients, not ATP. There are some um, channel proteins that are very specific for carrying certain ions, whether it's potassium or sodium, calcium, chloride. And so they actually have to get a stimulus from outside and say whether it's open or closed. Here's our carrier protein. And so we can see with our carrier protein, the high concentration, it's going to move it into the cell as that substance, or we can call that a ligand again, binds to that binding pocket, and then it changes shape and moves it to the interior of the cell. Now, we call these like facilitated transport because it's like they help. They're helping move the solutes into or ligands into the cell. Um, there are some things that really need to get into the cell, for example, glucose. Glucose, as you know, is hydrophilic, right? It's dissolved in water, so it cannot pass through the membrane without help. Same with certain amino acids. So those carrier proteins are going to help. So we have our sugar. It's going to bind. Let's see if I can move that real quick. See how it's empty? It's going to bind to that pocket. That's going to change shape, and it's going to bring the glucose in there. It is still going to move according to concentration gradient. Now, with glucose being so important for our cells, that is what we're going to make ATP out of. As soon as glucose gets in here, we're gonna put a high energy phosphate group on that. We're like, tag, you're not leaving. Because now it is not glucose, it is glucose. Actually, they'll put two glucose 1,6 biphosphate. It's a different substance. So we can pull that out of our concentration gradient for glucose because it is no longer glucose. Concentration gradients are very specific to that one particular molecule. Okay. Here is one of those ion channels. Oh, you guys hear about GABA, 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 right? So we have this protein here. This protein has 4 binding sites on it. If you can see here, it would be more of like a quaternary structure with multiple polypeptide subunits together to function as a whole. So we're going to have this chemical that is going to bind here. And then once we have our GABA bind to these two, it is then going to allow chloride ions to go into the cell. As those chloride items or as these chloride ions move with this stimulus, this is called the synaptic cleft. This is your nerve system. This is the space between your nerves. All right. This signal can stimulate an electrical signal or a chemical signal to pass information through your nervous system. All right, so that is it for this part of the 7.3, 7.4 part. I think it was 7.3, 7.4. I look at my book, but 7.1, 7.2, yeah. And then I'm going to do the now I'm questioning myself. The rest of the chapter in, yeah, 7.3, 7.4, in the next presentation. I'm just keep chugging these out, guys. Thanks for being patient with me, and I will see you in the next video. Bye.